Today we will discuss about this high spatial resolution multi-omics sequencing via deterministic barcoding in tissue. So in short term, this technology is known as DBiT. So it will provide us the information about spatial distribution of the genes that are present within the tissue. So without wasting uh, any more time, let's just go into the design and validation of this DBiT. So this is the tissue slide that is the tissue is fixed on a glass slide, and they have did the study on two different approaches. First, they have stained the tissues with the some of the identified antibodies or some of the well-known proteins, antibodies that are present against the well-known proteins, and thereafter they did this first PDMS or the first barcoding in this horizontal direction and the next barcoding in the vertical direction. So now they will have each pixels that are barcoded with different combination of the A1 and B1. Each pixels has the different combinations of A1 and B1. So thereafter what they did is they lysed the tissue and did the NGS library preparations, or collect the DNA and did the NGS library preparations, and from the NGS analysis they can see that what are the mRNAs and proteins that are present in the tissues. So, and in this technology they are able to identify greater than 22,500 genes or proteins. Panel they have used in this paper is about 22 proteins, but they claim that it can be used for 100 proteins. So, and this is how their instrument looks like. That is the PDMS slides, their PDMS slab on the tissue, whole embryo tissue looks like. And in order to validate whether there is a leakage in the channels, they use the Psi3 labeled barcode channels, and they can see that there is no leakage in the channels. And for the cross flow they used this FITC labeled barcode. So this FITC labeled barcode will only ligate to the position where there is Psi3 label barcodes are present, because Psi3 label barcodes contain the linker on which this FITC label barcode B can bind. So the FITC label barcodes are present on a square shapes, that only FITC channels we can see that shape. So how this ligations occurs, we can understand these things through this simple diagram. So in this diagram, so we can see that the mRNAs are usually present within the cells that contains this poly-A tails that is used to stabilize the mRNAs. So using these characteristics of the mRNA, barcodes are designed that contains poly-T tails and the barcode and also the ligation linkers. And since this tissue slice is already stained with 22 different antibodies against 22 different proteins, and each antibodies has different barcode, and it also has this poly-A tails. So these antibodies will bind to the protein, and this poly-A tails will bind to this barcode A. And after reverse transcription that occurs in this first step, so what we can get is we can get the mRNA sequence that contains this barcode and also the ligation linker. And for these antibodies we will get, for the proteins we will get this antibody barcode with this antibody barcode and also this barcode A with ligation linker. And after that, when this barcode B is used, that contains this ligation linker and the barcode B and this unique molecule identifier and the PCR handle with the biotin. Biotin they used so that it can be separated using, so that the cDNA can be separated in the later stage of their sample preparation. So after this second PDMS ligations, how the whole sequence will look like. It has this mRNA with barcode A, the barcode B, and this unique molecular identifier and the PCR handle, which is essential for the NGS sample preparation. And the antibodies contains this antibody barcode and the barcode A with barcode B, unique molecular identifier, and PCR handle. So, and so this is about how the uh, these barcodes are attached to each other. And now, so this is the in this panel C, they have validated the figure. They have validated the leakage in the tissue slide. However, in order to validate whether there is a leakage on the more rough surface, what they did is they grow the cells on the glass slide, and this time they use these HUVEC cells, that is endothelial cells, and then stain the cells with one side they stain the cells with DBiT, and the other cells they stain the cells uh, that can they stain the cells they stain the cell junctions. So we can see the cell junctions, and they can see that in the when they are staining the DBiT, even the DBiT is used to stain nucleus. So we can see that only the region that comes in this DBiT staining, only the region that comes in the DBiT stain channel is only gets stained, and the rest of the part is not stained. And similarly for the this cell adhesions linkage also, we can see that only the region that comes in this that comes in the vertical directions get linked, and the region that's not in the channel does not get stained. So this is this further validate their leakage of there is very negligible leakage in the DBiT barcoding. And after that, they further decided to check whether there is uh, a leakage within the whether there is a leakage in the Z directions or not. So for this, they did the confocal images, and when they check the signals in the Z directions, they can see that there is also no leakage they have found in the Z directions. And these images are for this fluorescence mRNA fish, that is fluorescence in situ hybridization. And in this case, we can see that there are some leakage in this when they use fluorescence in situ hybridizations method. However, in the case of this DBiT, they did not see any that much leakage that they see in this fish method, fluorescence in situ hybridization method. And when they quantify their leakages, or they can see that when they use this 10 micrometer channel, they can see around 0.9 to 2 micrometer leakage, and when they use 50 micrometer channel, they can see around 10 micrometer leakages. And similarly for 25 micrometer, they can see the leakages as around 1.7 micrometer. Leakages around 1.57 micrometer, right? So this is about the validation of the DBiT. And also when they compared their results, DBiT results, with the other established technologies, they can see that for the DBiT of this 10 with the 50 micrometer, the number of genes per pixels are higher compared to the other technologies such as ST, Visium, Slide-seq. And even for the 10 micrometer channel, they can see the high number of genes per spot. And similarly for this unique molecular identifier also, they can see the high number of UMIs for per even in this 50 micrometer channel and also in 10 micrometer channels are more than the other established technologies such as ST, Visium, Slide C. Next, they use this after validating their DBiT, they use this strategy to find out some insight about the mouse embryos. So what they did is they used the different adjacent slide of mouse embryo, and in the one slide they stain the mouse embryo with this HNE, and this embryo is at this E10 stage. And these are the next is the result of this mRNA mapping that they get from DBiT, and this protein mapping, they use 22 different proteins that they get from the DBiT, and thereafter they compared their uh, different mouse embryos results with this bulk RNA-seq data with the RNA-seq data of the RNA-seq data of the other mouse embryos at other stages with this DBiT data, and they can see that the other RNA pseudo bulk RNA-seq data, that is RNA-seq data for the E9.5, and this E10 pseudo bulk RNA-seq data of the E10 lies between the RNA-seq data of E9.5 and E10.5. So this further substantiate the point that their results from the DBiT are comparables to the other RNA-seq data. And thereafter they cluster, they use this UMAP clustering to cluster the genes that they have identified from the DBiT, and after clustering them they again do the spatial clustering, that is each clusters genes that are present in this cluster will again be spread across this spatial clustering, and they overlap spatial clustering with HNE that can be seen here. And from the literature they identified this different 11 clusters belongs to this kind of groups, or they have this based on the common things that are found in this cluster. They identified that these clusters belongs to this part of the mouse embryo, that is the first cluster belongs to this disenfal development, and the second cluster belongs to this forebrain development, and etc. And after doing this clustering they again do a, based on the literature study, they did the manual clustering, and which has this 11 different notations. And this manual clustering, uh, most of this manual clustering overlap with this spatial clustering that they obtained from the UMAP unsupervised UMAP clustering. And after that they compare the mRNAs and protein expressions, some of the mRNAs and protein expressions in the different cluster, and they can see that most of the mRNAs and protein expressions results are comparables in the different clusters that can be seen here. If the mRNAs expressions are high, then the protein expressions are also high. So this also validate the DBiT results, that this also validates DBiT results. Next, after doing the whole embryo, they decided to check spatial organs, or they next focused on the spatial or specific organ of the mouse. So they focus on this brain of the mouse using 50 micrometer channel. So this is the mRNA mapping, and these are the protein mapping that can be seen here. And this is the HNE image, HNE staining image. So in this case what they did is they have mentioned about the 4 different proteins and the expression labels spatially, that is CD63, pan-EC antigen, MATCAM, and CD26. And this time they compare or they quantify two of the results, that is EPCAM results and this pan-EC results through through immunostaining of they compare the results that they obtain from DBiT about EPCAM and pan-EC antigen with the immunostaining results that they obtain from the neighboring tissue slides. So these images are the overlap of the DBiT spatial clustering of this EPCAM and also the immunostainings. And this is the comparisons of the here the EPCAM immunostaining results based on the intensity and this EPCAM DBiT results based on the UMI count. We can see the results are comparable for the EPCAM and also for the PECA. Some deviations, they said that these deviations might be because of the different tissue slides that they have used, as the same tissue slides cannot be used for both the studies. And thereafter they cluster this whole results. They cluster the results from the DBiT in different into 10 clusters, and then they do the spatial transcript. They cluster the transcriptomes that they obtain from the DBiT into different clusters, and when they do the spatial transcriptomes, when they do the spatial clustering, and into these 4 different groups have been identified. And we can see that this cluster 2, which belongs to this blood vessels network and red plus vessels, based on the literatures comparisons to the genes that are present in the cluster 2, they can see that this cluster 2 looks similar to this pan-EC antigen, and pan-EC antigens proteins are also belongs to this categories, that is blood vessel network and red blood vessels. So they further, from these results, they further substantiate their point that pan-EC antigens is their DBiT technology is more powerful. It's powerful. Next, they did spatial gene mapping of the early eye development, or when the eye development is at the initial stage. So for this they use 10 micrometer channel, and they use this section, this red section, for the DBiT or spatial mapping. This is the zoom in image of this red section, and this is the H&E image of the red section. And after this DBiT results, they use the 4 important genes, that is PMAL, NPEC6, NTR, PM1 and SIX6, and from there they can get the idea about this optic vesicles formations. And in order to compare these results with the whether it is early development stage of the eye or not, they also did the DBiT on the next stage of the mouse embryo, that is at E11 stage. And from there they can compare that at E10 stage cells is at optic vesicles, or this eye is at optic vesicle stage. However, at E11 stage the eyes move into this optic curve stage. And similarly they use the other genes also, such as PEC6 also, and this 6 also shows the similar results. And apart from this, they also check this for other markers for the eyes, and they can see that for E10 stage these markers are can be seen. However, at the E11 stage these markers are very less. And these results are also in line with the literatures, that is well published. So far they compared the results, and it is well in line with the literature's DBiT results. Next, they decided to couple their DBiT with the single cell RNA-seq data, data. So for this, what they did is they have the results for the DBiT uh, for this E10 stage, and they use the E9.5 single cell RNA-seq data and E10.5 single cell RNA-seq data, and do this UMAP clustering. And what they find out is that this red dot, this data are easily grouped among this RNA-seq data, and the red dots are the DBiT data, and this gray and blue dots are the RNA-seq data, single cell RNA-seq data. And they, when they do the clustering of this UMAP, they are able to cluster them in 24 different colonies. And when they did the when based on the genes, they are able to group them in like around 54. I'm not able to recall the exact name, but around 54 groups. And this is the UMAP clustering is then again converted into this spatial clustering. And one thing, so after doing this identifications, what they did is they used they took some of the clusters, such as this A, that they have defined it as A cluster. And when they do the spatial clustering of this, they can see the different parts of the eye development, that is retinal epithelium development and retina trajectory. So this 10 micrometer channel, with 10 micrometer channel, they can able to see different kinds of, able to distinguish different parts of the eye development. And they did not only for this A part, but also for this B part that has the different clusters. B part has like these clusters, 3 clusters. So on the B part also we can see that these endothelials and erythroid clusters are formed, and respectively they also did D, C, E, F imaging, and they can find the different part of the eye development. It is connective tissue, epithelium tissue, muscle cells. And so from their 10 micrometer channel, this DBiT, one can able to find out, even able to distinguish the organ development at the early stage. So this further shows that how powerful these DBiT technologies are. Next, thereafter they do this global clustering analysis of the 11 mouse embryos. So they took 11 different mouse samples at different stages, that is E10, E11, E12, and here is the sample list. And when they cluster them, they are able to cluster them into the 20 different groups using Tisney, and these are based on the clusters, and this is based on the samples. And from the this literature analysis, they can group those clusters into the different developmental stages. That is, cluster 2 can be grouped to this muscle system processes. So this can be grouped to muscle system processes, and this is basically at this E11 stage. E11 and E11 tail stage and E11 tail. 10 micrometer channel and E11 at 25 micrometer channel. So and one thing that they can see that in this UMAP is that although different samples are clustered themselves into the same group, however for this channel, that is E11, for this E11 channel they can see that E11 channel is not grouped together into the single cluster, however it is grouped together into the 4 different clusters that can be seen here. So they decided to check why this is clubbed into the different channels. It might be because of the tissues or samples are different. Tissues are has a very high heterogeneity. So they further checked why these clusters are different from each other, and along with that they also see that the 50 micrometer channels UMAP varies significantly from the early stage to the later stage. So this indicates that 50 micrometer channels has more heterogeneity at different stages, and this is also in line with the general hypothesis, as 50 micrometer channels contains more number of cells. So there is high possibility that different stages of the mouse has different expressions of the genes within the cells. So in this figure they mapped why these clusters are different from each other. So when they map this cluster into the spatial form, they can see that these clusters are represent these spatial forms, and this and they are able to this E11 embryo lower body are able to further cluster these ABCDs are able to further cluster themselves into this 12 clustered, and thereafter they do the spatial clustering that can be seen here. And when they did the group based on their literature's data, annotate this based on the scRNA's data, they can see that it can be divided into these uh, different trajectories, and finally what they conclude that these groups that they are clustered are belongs to the different internal organs based on the comparison between this RNA-seq data and this E11 embryo body. That is this, for example, this A part belongs to this hepatocyte trajectory, and from which organ this belongs to this hepatocyte trajectory they find out, and this A part belongs to this erythrocyte coagulations. Similarly, they compared these results that this neural tube belongs to this uh, part B, and this ventricle belongs to this part C, and simultaneously other parts are been mapped. And after mapping this part, they also checked the markers of the genes belongs to the different organs, and they can locate the different organs based on their markers. For example, liver specific markers such as this My6, APOA2 and erythrocyte coagulations markers. So they can predict where the internal organs are developed at different stage of the mouse embryo. So this is about how this DBiT technology can be used to get more insight about the spatial distributions of the cells. and it can even be clubbed to the single cell RNA-seq technologies, and together we can get more insights about the changes that are occurs within the cells because of the spatial distributions, and which can improve our knowledge about this biology to a greater extent, and it can help us to develop the therapies that can be more effective and will be able to cure the disease that are presently uncurable. So that's all for this video, and if you need to understand more about this DBiT technology in detail, you can go through the reference that has been provided in the description. Thank you for your time.